All right, thank you very much. We had a couple of fantastic talks, and now it's my turn to tell you about how we've been harnessing large, ultra-large chemical compound data sets to improve our digital chemistry pipeline. And so most people here probably already know how complex the problem of drug discovery is. The classical way that we used to find new drugs relied heavily on basically artisanal expertise of medicinal chemists to basically find almost manually these compounds. Of course, this is not scalable. It's extremely costly. And because of this, in the past 20 or so years, there's been a real push in industry to try and fix this issue by automatizing as many elements as possible of the drug discovery pipeline. Luckily for us, we've now reached a critical mass in terms of data availability, especially in terms of these chemical compound libraries that vendors are offering for synthesis. And this allows us to finally see some interesting results when applying these very fascinating ML and AI models to explore these spaces, find new drugs. But we are trying to use these spaces really not only to find new drugs, but actually to find new types of drugs. And I want to stress this point because the goal here is not really just to keep going and screen larger and larger libraries. That's just not feasible. The goal here is to use these as tools to try and understand which regions of the chemical space we are currently missing, and try then to build better tools to explore these unexplored areas of the chemical space. We do believe that there's some treasures in there that we need to extract. And so to do that, we need the right tools. If you think about, how do you design a model to traverse these spaces and find new drugs among all these compounds, you can reduce this problem to a simple question. Given a compound, does it bind, does it interact with my target of interest, with the protein I want to study? Yes or no? Of course, in an ideal world, we would like to know as much as possible about the kind of interaction we are looking at. We would like to know how strong this interaction is and what kind of bonds the molecule forms when attaching itself to the surface of the protein. So people, of course, jumped at that. And, if you look at literature, there are some very, very interesting examples of models that try to predict what is called the docking pose of these molecules. So what kind of configuration these molecules take when binding to these proteins. They're really interesting from a scientific exploration perspective, but when we look at their performance on real case scenarios, the quality of the results, it's quite underwhelming, especially compared to what we can get with classical docking, which is a tool that we've been using since the 80s. And so the issue here is not as much with the models themselves. They're actually pretty smart models. The issue is with the data. We just don't have enough accurate data to build models that go so much into detail. And so we can maybe take a step back and go back to the question that we're trying to answer. Maybe we don't need to know everything. We just need to answer the question, does this ligand interact with this protein, yes or no? So drug target interaction models, or DTI models, do exactly this. These are supervised binary classification models that try to answer this one question, but try to do it well. Because they don't need so much information about the binding, we can actually leverage these a number of different databases like Campbell, BindingDB, that collect a lot of DTI information from experiments and papers and literature, and make it publicly available for us. And so we can use these, and if we put them together, we actually have enough data to build models that perform decently well. Of course, we are not the first ones that thought about building a DTI model. They're actually quite popular in literature in the last 5 or so years. I'm putting here a non-exhaustive list of models that are published in literature. You can see that the list stops at 2022. I wanted to update it for this presentation, and I went on Google Scholar. I searched for DTI models, and I had to give up. So I just stopped it here. That's enough. It's just not exhaustive, but there's many more. So the issue, I mean, of course, being very interesting for academia, especially, the methods are quite diverse among these different tools that are published. And of course, there's also a bit of a bias in terms of the interest. Most of these publications focus on building different architectures. You know, neural networks are cool, and everyone wants to build the new interesting architecture. Nobody really wants to think about how to clean the data, how to pre-process the data, and all those homework almost that we need to do to actually train proper models. And so not much thought has been put into how do you handle negative data. Most of the DTI information that is available is positive data, because not many experiments are really run on molecules that are known not to bind to their target. But most importantly, also, not many people think about how to properly test these models to make sure that the performance we measure actually reflects how they behave on real data. And so another major issue with some of these models is that they're actually target specific. They only take ligand information, and they can tell you if these ligands interact with one target or maybe a family of target. But this is just not manageable for a drug discovery pipeline. We want to be able to run, we can't just build one model for every target we have to work with. We need a model that is able to predict DTIs across a vast variety of proteins. And even more importantly, it's generalizability. We want this model to be able to run inference on new targets, not only to recapitulate the knowledge that it learned from training. And so MatchMaker is our DTI model that tries to answer this question. So it's a deep learning DTI model. As I just mentioned, it's a binary classifier. It's trained on 3.4 million ligand protein pairs for around 1,700 species. We took care of making sure that there was a good variety in the protein space. It takes information both from ligand representation, so fingerprints or physicochemical descriptors, but also from the proteins themselves. So it needs to look at the structures, either from experimentally available crystal structures, or when not available, from AlphaFold 2 models. And again, we put a lot of thought in how do we filter the data, how do we handle the negatives, and also how do we properly test this model to make sure that it's generalizable enough to be used within a drug discovery pipeline. And the good thing of having a model that actually looks at both the compound and the protein is that then you can extend the screening to a large set of proteins and not just limit it to one specific target. Of course, we want to use this to find new drugs to fix human diseases. So the human proteome is the major set of proteins that we look at. There are around 60,000 proteins that have structures available among those of the human proteome. But on top of that, some of these proteins come with different pockets that can interact with different ligands. So in truth, when we run the proteome screening, we run it on 80,000 different pockets coming from these 16,000 proteins. So what's the advantage of running a proteome screening? Well, first of all, we're not limiting ourselves to known targets. So we can have a look at all of the proteins of the human proteome and identify if our compounds interact with proteins that maybe were thought to be undruggable. The second point, and Marisa mentioned this before, we care a lot about polypharmacology. If you are, if you have a compound that you design to interact with a specific target, you want to make sure that it's not interacting with other targets, especially if these other proteins are involved in biological pathways that lead to toxicity or side effects. These are important questions that we want to ask ourselves when we design these models for a drug discovery pipeline.
Before you go off from that slide, I just want to make sure for the software engineers in here, that's what we expect you to do.
Yeah, yeah, fair enough. All right. So of course we build this model and we run it with some with good success, with some partnership, but then we wanted to put it to the test to explore one of these ultra large spaces. So Enamine is a chemical company, is one of the most famous chemical companies because it offers a wide varieties of small molecular weight compounds that are very useful for drug discovery. And if you go on their website, you will see they offer a number of different screening libraries. The most popular one is around 4 million compounds, and the largest one contains around 6 billion compounds, so 10 to the power of 9. However, you can actually order off menu. Oh, sorry. The actual full space that is accessible to Enamine based on the reagents and reactions they have access to is actually 6 times larger. It's 36 billion total molecules. But if you try to access the space, if you ask them, can you give me the molecules, what you will get is instead is a list of 250 symptoms and a manual of composition rules. Just to clarify, symptoms do not map to actual reagents that Enamine has access to. Those are trade secrets. We don't have access to those. But there are these sort of modular chemical structures that are quite useful from a theoretical perspective to understand the structure of these compounds. And you can think of them as sort of Lego bricks of chemistry. So they are quite useful to understand what's going on, but they don't represent the actual reactions that they have access to. And so they gave us this list, they gave us the rules to put these Lego bricks together, and they told us, figure it out. And that's what we did. So we enumerated all of the possible combinations of these symptoms with the composition rules, and we recovered the 36 billion compounds that they can synthesize for us. And after that, of course, we need to extract the physical-chemical properties that MatchMaker can digest to make its predictions. So here on the right, I'm showing you a lower dimensionality representation of what this physical-chemical space of the 36 billion looks like as green dots. I'm actually cheating here. This is only 1 million dots because I just couldn't fit them all in for various reasons. But it's representative, so it's pretty close. The reason why I'm showing you this, I don't know if you can see, I'm also showing some red dots, and that's essentially the representative of a typical screening library of 4 million compounds, as I mentioned before. So the red dots is what we are usually using to run our screening. And the green dots, you can see how much space in this chemistry landscape we are effectively missing on. Of course, not all of these compounds are useful, but it's guaranteed that there's very important, very interesting chemistry going on that we are missing out. On top of that, also, screening libraries come with some biases, and working with a full chemical space actually allows us to go beyond those. And so we finally took these 36 billion compounds and we ran them through MatchMaker. And so some of you may have seen the announcement we made a couple of months ago. We ran the screening of the 36 billion compounds against our 80,000 and more pockets, and that gives us 2.8 quadrillion, that's 10 to the power of 15 pairwise interactions. And this was possible only thanks to our collaboration with NVIDIA, who gave us access to their cloud system, and also thanks to what I can only call an abuse of BioBytes, our supercomputer. And yeah, so 2.8 quadrillion, that's a lot of data. So I added this slide to put things into perspective a little bit to make you understand how much data we're talking about. So if I wanted to show you all of the hits that we found in a plot like this, a sort of heat map, showing all the proteins, not even the pockets, just the proteins on the x-axis, and on the y-axis, all the compounds. And if I were then to make this plot smaller, such as that those squares are as small as a pixel, sorry about that, I will need a screen that is as large as a hippopotamus is long. I don't know if these things into perspectives. And also I realize now that this screen is actually pretty big, so it's not that impressive, but when I was thinking about this, it looked impressive in my mind. Anyway, it's a lot. Hippos are massive. So more impressive is actually the fact that I will need the screen to be as tall as to reach out to the external layer of the exosphere. So we are talking about 10,000 kilometers, so 6,500 miles for the Americans in the room, which is the majority, I believe. So it's a lot of data. So the next question is, how do we deal with this? So much data comes with a lot of challenges. From the technical point of view, it required a lot of compute resources to get this data out. Even just storing the raw results, even before analyzing them, we need a lot of space, more than 1 petabyte of space. And so even just parsing, sorry about that, all this space requires, it's quite slow. It requires a lot of effort. And on top of that, then come the scientific challenges. How do we prioritize these compounds? How do we interpret the finding? And yes, this space may be great for inside mining, but we can't really then run experiments on all of the single compounds that come out of this. And so while we think about the logistics of dealing with the 36 billions, however, we should also remember that we also have the space that we can use. And this is actually a surprisingly tool for us. I was really skeptical at the beginning when we started looking at symptoms. It does reduce the problem to only 250K data points. Plus the symptoms are being these very essential building blocks, working on structure activity relationship, trying and understand how they behave based on the structure is actually a little bit easier than working with complex compounds. But most importantly, we observe that even starting by very simple statistics in the 36 billion space, summing, averaging our results, and then mapping them back onto the space of the symptoms gave rise to these very rugged, very complex maps, very entropic maps. And that's basically telling us that there's a lot of information that we can actually mine from this space that is, however, a bit more manageable to work with. And as you map this information into distributions, you can do then a lot of neat stuff. You can look at population statistics, enrichment. You can look for families of proteins and see if they have a preference for certain compounds containing certain symptoms. And this basically, it's a good step towards our chance to finally understand the mechanism of action behind the binding of these compounds with these proteins. And so I wish I could share a little bit more. You need to understand that this is fresh out of the oven. We still didn't even have the chance to sink our teeth in this data, and we are quite excited to see what can come out of it. However, we are already thinking about how do we use these data to enhance our digital chemistry pipeline in the first place. So we can use this predictor score, this predicted interaction to prioritize wet lab experiments. We don't really need to run experiments on every single element, every single compound in the library. And that doesn't mean that also this is the last tool that we need to use. This could be a pre-screening filter. We identify hits here, and then we can use more computational intensive, but also more accurate tools like FEP, sorry about that, to actually refine our hits. And most importantly, this is really just one of many layers that Recursion is building as part of this multimodal dataset, multimodal maps that we're trying to use to understand the chemistry and biology behind this binding. And so I want to reiterate the idea that we're not really just screening these libraries for the sake of it. Yes, they're really difficult to work with. There's a lot of technical challenges ahead of us, but we really truly believe that it's worth it. There's a lot of chemistry, a lot of unexplored space and unexplored drugs that we can finally find within these spaces. And the amine synthesizable space is just the beginning. And we believe that, there's a lot of cool stuff that will come out of these analyses. And so I have actually a lot of people to thank. A lot of professional people worked on this project. I couldn't put all of them in these slides, but I put a couple of faces up there. And so thank you everyone who helped out with this project. And yeah, thank you. Oh, okay, there was supposed to be a thank you there. Oh, no, wait, here it is. Okay. was scared for a minute, but yeah, okay. Yeah, so thank you. Questions?
Can I ask a question? I don't know if you can hear me. Okay. So what's the difference between the synthons and like chemical scaffolds? Like, are they the same, or is synthon specific to the enamine data set?
No, so symptoms are not specific on enamine. So it's a bit tough in the sense that it's not very well defined how symptoms are. They are not necessarily scaffolds. It depends how you define them, but they're more like very, very basic structural components that you can, from which you can reconstruct the chemical compounds by simply substituting, for example, one atom with another syntone. So they're very abstract and very basic elements. They cannot necessarily be used to grow a molecule out of, for example, like you would for certain scaffolds. So they don't follow the same requirements that you would have to define a scaffold. They're very abstract, but they're not, enamine didn't invent them. They're just used sometimes because they simplify things. But yeah.
Okay. And just, sorry, one follow-up question. So they're like kind of abstract in the sense of maybe like they have no chemical nature property in and of themselves, but they are like the puzzle pieces to put them together, and that's how you were discovering the space instead of using like a scaffold that's like a more quote unquote formal structure.
Pretty much, yeah. So some of these may not actually exist in the real world as chemical entities. They come with, for example, wildcards. So atoms that are supposed to be substituted with other pieces of chemistry, of chemical compounds and chemical structures. So yes, pretty much correct. Yeah. Thank you. Yeah. I have a background in physics, actually, and I had to learn my chemistry as I went, pretty much, but yeah. So my path was a bit weird. So I started studying physics, then I moved on to atomistic simulation of biosystems, like molecular dynamics and stuff like that. So that exposed me to a lot of biology. But still, it was a very physics approach to things. You're effectively simulating your atoms as spheres, interacting with springs and stuff like that. So it's still a very dry approach to biology. And then I moved on to actually work on genomics. And then I started interacting with a lot of biologists. And that also exposed me a little bit more to, this kind of concept. So I sort of had to catch up as I go. But personally, from my experience, I appreciated a lot working in these environments with, people from different backgrounds, different expertises. We were complementing each other a lot. I had colleagues. I was teaching them about statistics. They were teaching them about biology. Incredibly smart people, and I appreciate a lot places that can recognize these and put together teams with different skills. It really does work. We are more than the sum of our parts. If you put together people who have this different expertise, you can actually access, something more. So yeah, this is it. Yeah. Yeah. I don't know, maybe I went a bit too personal. Sorry. I don't know.
I can't hear if anyone's asking a question. Can I ask one more? Sorry.
Yeah, no. Do you want?
Yeah, you just started.
It's okay. Yeah, sorry, you had another question online?
Yeah, Evan, me?
Yeah, sorry.
Okay. Yeah, sorry. This is my last question. I've thought a lot about this concept of synthons. I've never had a word for it. So I really have enjoyed this talk. But is it possible that you could begin to like, quote unquote, I guess that's what you were saying, but like link synthons to like specific like sections of a protein's pocket or an active region and begin to find patterns that way. So then across proteins, you see like, oh, well, during this like part of a protein pocket that like, let's say across like kinases, you know, they have this hinge pocket, like if you begin to see a pattern kind of like that.
Yeah, so that's pretty much the idea of when I was showing you
Okay.
on the symptoms. The idea is, you know, you can take even just the average of these predictions and say, okay, kinases really prefer compounds that contain this type of symptom. We need, however, to be a little bit careful because, you know, proteins are really complex systems. And, you know, yes, maybe you can say this family of proteins prefer to have, you know, an amine in this region or something like that. But the hard truth is that more often than not, the binding actually requires all of the elements of the ligand to work with each other. There's a category of binding pockets. Sorry if I'm a bit going, you know, wild with this, but there's a category of binding pockets which are called cryptic binding pockets, which are generally not apparent in crystal structures because they actually require some very complex set of interactions with the ligand to open up the pocket. So the ligand needs to get there, then it moves stuffs apart and the pocket opens up and the ligand gets in. So those are very, very difficult to work with, but it gives you a little idea of the complexity. Like you might not be able to completely understand how the binding works if you're really working with just partial information, but you can still get some information out. So we do hope to abstract some chemistry with these symptoms, but we are not just going to work with them. It's just one among many tools, essentially.
Yeah, that makes sense. I totally agree. And yeah, the cryptic pocket, that's, it wasn't on my mind, but that makes sense as well. So thank you.
Thank you. Great question. Yeah. Well, yeah. So there's a lot of layers to it. Okay, you have phenomics that tells you about, you know, how the cell look like and how the final product of, you know, knockouts and these compounds interaction look like. You have transcriptomics, you have proteomics. So there's a lot of actual layers in this, you know, this kind of vision of biology. And chemistry is just one of them, one step among all of these. We would like to get to the point where we can actually complete the loop as you defined it, okay? At the moment, we can still work with what we have. We can, you know, look at the interaction that we predict from the chemical point of view and then try to cross-reference them with what we observe in our phenomics maps. But we need to be aware that we are looking at very different, you know, perspective. We're looking at the same thing, but from different perspectives. There's so many elements that go into the phenotype at the end. Often you will not see connection with the chemistry because you're missing the link in between, essentially. Biological pathways are so interconnected and so complicated. It's not easy. So we hope eventually to, to connect the loop. That's the plan, but it's gonna take a lot of work. So for now, we are trying what we can with what we have, and we believe that we can still get a lot of cool science out of it. But yeah, there's a lot of work ahead of us, definitely. So yeah, great point. Yeah, thank you. All right, that's it. All right, thank you.